Okay, let's talk about biological buffers. So buffers are really important to protect the integrity of biological molecules. So biological molecules are what? The molecules that are normally made and found from living cells, right? The examples are what? Proteins, DNA, RNA, and carbohydrates or lipids and things like that. So since your body maintains a constant condition, okay, so the pH level within your body is constant, salt concentration is constant, so those biological molecules maintain their structure under that constant condition, okay. But often in the biotech industry, you are dealing with those biological molecules outside of the living cell or living system. Think about it. Genetically modified CHO cells are grown up in the bioreactor. At the end, they will be released, those CHO cells, their API, maybe monoclonal antibody, from their cells to outside medium. And then once those proteins are released to outside of a cell, they are very prone to, changes. Okay, like the pH changes, the fluctuation of hydrogen ions, okay, the salt changes, which can break up their, ionic bonds and things like that. So they will change the integrity of biological molecules. So if you want to deal with those biological molecules outside of a cell, for example, in vitro, inside of a test tube, you have to provide a certain constant condition where pH changes will be minimized or salt concentration will be minimized. Among those different things, of course, the pH change will be minimized through the function of buffers. Okay, so lab buffers are the lab solutions that help maintain biological system at the proper pH. Normally it's around like a neutral pH area, because biological buffers can suck up extra hydrogen ions, and by doing so they can resist the sudden pH change. That's what they do. So the pKa value is a certain pH range where buffers will resist a sudden pH change by binding with the hydrogen ions. Okay, for example, acetic acid, can protect the pH change around what? pH 4.8. If you add more NaOH or add more hydrochloric acid, the change will be very slow because the acetic acid can bind a lot of those, so it resists the pH change. That's what buffers do. By doing so, they protect biological molecules that are existing in the solution together. That's the function of the buffer. Okay. So buffer themselves should be inert, meaning non-reactive. Okay, they should not react upon your API or biological molecules that you are trying to isolate or purify. Right? So buffer, their main function is to protect your molecules from pH change. So they should not react with your target molecule, of course. So examples are these buffers you may have made in your lab, TBE or TBAE, electrophoresis buffers. Depends on what acid you add to tris to adjust the pH. They would become either TBE or TAE. Depends on did you add boric acid or acetic acid, right? But between those two, actually TBE is reactive to the DNA molecule, so that's why you use TAE if you're going to purify your DNA after the electrophoresis is done, because your DNA may lose its integrity. Okay, anyway, buffers are sensitive to temperature change. Of course, temperature affects many different things. Among those, the buffer pKa value should be also affected. So you need to keep the constant temperature. That's important for your buffer to be work functionally. And then buffer themselves can be adjusted in their pH. For example, tris base is a strong base. If you dissolve trisma base in water, its pH value will be above 10, so it's a strong base. But normally the buffering capacity of pH is around like a neutral, maybe 6.8 or 7. So then how do you bring down its pH? By adding some acid, like hydrochloric acid. You can bring down its pH. Keep the the needed pH with this buffer. That's how you do it. So hydrochloric acid, when it's added to tris, tris becomes a tris Cl because hydrogen ions will be binding with the tris. So that's the pH adjusted tris. Okay. Sometimes there are chemicals that comes in monobasic and dibasic forms, And in that case, you can mix those together, monobasic or dibasic forms of these chemicals. By doing so, you can adjust the pH of these solutions, like sodium phosphate buffer, for example. There is a monobasic form and dibasic form. Monobasic form is a little acidic, dibasic form is of course a little basic. If you combine these two in certain ratio, you can adjust the combined solution and it's going to work as a buffer. Okay. So again, why is it important to provide buffer in the solution that keeps your biological molecule? Let's give you an example. So you're dealing with the protein molecule. You know, a lot of API molecules are made up of protein, right? So let's review what proteins are. Okay, so monoclonal antibody is a protein molecule. You know, insulin is a protein molecule. Or what else? The peptides are normally protein molecule. There are so many API that's protein based. Then proteins are what? Made up with amino acids, right? So the building blocks of proteins are amino acids. So simplified chemical equation of amino acid looks like that. There's alpha carbon in the middle, side chain, amino group, carboxy group, and hydrogen. Okay, there's one amino acid, there's another amino acid. If you put them together, there's a peptide bond formation. That's a strong covalent bond. You can, form it. Then once you form it, it's not easily broken up. So that's how you make a protein. Okay. And there are 20 different amino acids, as you know. So these are 20 different amino acids. Among those 20 different amino acids, there are so-called hydrophobic amino acids. If you look at the side chain of these amino acids, they are non-polar, hydrophobic. Okay, because of that, these amino acids are considered hydrophobic. And there are also hydrophilic amino acids. If you can look at the structure of the R group here, OH minus, it's hydrophilic, it's water-like, right? Polar, strongly. And these guys are also hydrophilic. So there are hydrophobic groups, hydrophilic groups, as well as what? The basic and acidic groups. So a couple of acidic amino acids, aspartic acid, glutamic acid, as well as 3 of the basic amino acids. So there are about what? 4 different amino acid groups that are out there. Okay, and depends on what amino acids are making your proteins. Okay, through all those peptide bonds, your protein's size and how many of them are making your protein, and then the structure of the protein will be determined, because all these amino acids, when they are forming a protein molecule, polypeptides, not only they can form the peptide bonds, they can also form what? Ionic bonds, or hydrogen bonds, or hydrophobic interactions, or van der Waals interaction, all those non-covalent bonds as well. So based on their non-covalent bond formation capability, your protein will fold up in different ways. Okay, so primary structure is based on the peptide bonds among all those amino acids. But hydrogen bonds, if they are involved in this amino and carboxy group of your peptides, then you will form the secondary structure, like beta pleated sheath structure or alpha helix structure. Those are secondary structures based on the hydrogen bonds formed among different amino acids. And then there is a so-called tertiary structure of your proteins, which is based on what? The side chains forming different types of interactions, like hydrophobic interaction, hydrogen bond interactions among those side chains, or maybe ionic interaction between acidic and basic ones, and van der Waals interaction. All those non-covalent bonds, if they are formed among those R side chains, you can see protein structure is changed dramatically. So it may develop some kind of a three-dimensional globular structure. That's a tertiary structure. And then if your protein is made up with multiple polypeptides, okay, then you consider that as quaternary structure, of course, right? So quaternary structure, how is it formed? Again, through those non-covalent bonds between those multiple peptide bonds. So these two polypeptides come together through non-covalent bonds. So those are the protein structure principles and how they fold up. Okay, so if your protein has a unique structure like this or like this, okay, so how do they maintain their unique structure? The secret is by maintaining all those non-covalent and covalent chemical bonds within them. That's the secret of their structural unity, uniqueness among those proteins, right? So proteins, their function is based on their unique structure. So structure equals the function, right? So if you make an active pharmaceutical ingredient that still maintains its activity, let's say you are making a monoclonal antibody which will bind to the antigen that's found from a tumor cell. Okay, so that antibody can do that because it has a fitting shape to that surface molecule on the tumor cell, right? So maintaining the antibody's unique structure is very important, okay, because that determines its function, right? So how do you keep or maintain the integrity of the structure? By providing the condition where, you know, all of the covalent, non-covalent bonds will be maintained, right? So all these bonds, first hydrogen bonds are formed between what? Hydrophilic groups, right? For example, water. If there's a single water molecule, it has a polarity, because, you know, here there's a bunch of protons, and there are a bunch of electrons that are kind of a dipole, okay? If we place another water molecule next to it, since water molecule also is dipole, there may be an interaction between dipoles, so negative electronegativity and positivity. That's the basis of hydrogen bonds, right? So yeah, proteins do have a bunch of hydrogen bonds, okay? And the hydrophobic interaction, if you have non-polar hydrophobic amino acid side chains, so non-polar amino acid, you know, they will fear water. They don't like to get around with water, so they will hide away from water. How do they that? By moving away from water and by clustering among themselves, hydrophobic ones together. So when they do that, you see very weak interaction among them. That's the hydrophobic interaction. So yes, proteins do have hydrophobic interaction. Mostly these amino acids that are hydrophobic are normally buried inside of a protein, okay, because they are hidden from water molecule. So that also is important for the protein molecules to maintain their structure and function, the integrity, right? So hydrogen bonds and hydrophobic interaction. And then between the acidic and basic amino acid side chains, you can form ionic bonds, right? So negatively charged amino acid side chain, positive charged amino acid side chain, they can interact by forming hydrogen bonds. And, you know, van der Waals interaction, of course, it's a transient movement based on, you know, dipole interaction. Just like hydrogen bond, it's even weaker than hydrogen bonds. So by combining all those interactions, non-covalent bonds and covalent bonds through the peptide bonds, your protein will maintain its structure and integrity. Okay, so your goal is to keep their structure integrity constant while you're working on those during your purification process of, you know, downstream or formulation. Your protein should maintain its structure and function. Okay, how do you do that? By providing a buffer or by providing the correct salt concentration and all those things, right? So here's a good question. How do proteins lose their structure and function? I told you, you know, their structure is based on all those covalent and non-covalent bonds. If those proteins are exposed to any condition that can compromise those non-covalent and covalent bonds, of course, proteins will lose their structure and lose their function. Okay, we call that denaturation process. If proteins lose their structure because a lot of the non-covalent bonds are broken up, that's just denaturation. You don't want that, right? So if your protein is exposed to high temperature, for example, high thermal energy, will break up a lot of those non-covalent bonds. And extreme pH changes like acidic pH or basic pH, they will also break up, you know, the hydrogen bonds as well. Ionic strength, if you put your protein in a high salt buffer or very low salt buffer, you know, all the ionic bonds will be broken up. So all those conditions are not good. Your protein should not be exposed to those denaturing conditions. And another way of breaking down your protein is enzymatic degradation. There are so many proteins that will be chopped up by different enzymes. We call the enzyme as protease enzyme. So enzyme has their name, typically ends with ESE or ASE. So that those enzymes, protein chewing enzyme is proteases. So those enzymes can catalyze the dipeptide formation. So breaking down peptide bonds, that is what these protease enzymes do. So if you expose your protein to the enzyme, yes, they will lose their functional structure. Oxidation is another way of losing protein structure. So your cysteine amino acids, there is SH group. Going back to the amino acid structure, there should be a cysteine amino acid somewhere. Where do you see that? Cysteine right here, right? SH. If you place a cysteine next to another cysteine, it's going to form disulfide bonds, right? Disulfide bonds will be formed under the oxidating condition. But if you place that protein to the reducing condition, that disulfide bonds will be broken up. So that's again not good. So oxidation or reducing condition is not good to maintain the integrity. Aggregation is another way of losing the protein structure, okay? So none of those are good at all, okay?